Okay, this is part 2 of section 10.2, and we'll be talking about mitosis and cytokinesis. Okay, so when we're looking at mitosis, there are 4 main events that happen, and they are prophase, metaphase, anaphase, and telophase. Okay? So the first phase of mitosis is called prophase. And now remember, in interphase we had the chromosomes where they duplicated, and they're called sister chromatids. Now in this case, here is where the duplicated chromosome condenses and becomes visible. So now this is where they'll look like the little X's that you're used to seeing. Those centrioles move to opposite sides of the nucleus, and they form what are called poles, and they help to organize that spindle. During metaphase, which is the second phase, those centromeres on the chromosomes line up across the center of the cell. It's very easy to tell metaphase even under a microscope. It's when the chromosomes are all lined up in the middle. All right? So those spindle fibers connect to the centromere of each chromosome, and they help it to line up along with the poles. Remember, the centrioles help to form the poles, and they line them up in the center of the cell. The third phase, which is called anaphase, this is when those poles start to turn. They turn and they actually pull the chromosomes apart. They pull them apart at the centromere. Okay? And these chromatids will separate, and now they would be individual chromosomes. The reason why they look kind of like a V like that is because it's supposed to be like if it's pulling apart. Now, in anaphase, the chromosomes will separate into 2 groups near the poles of the spindle. Okay, so they get, and they keep pulling them until they're close to the spindle. And then we go into telophase. And telophase is the 4th and final phase of mitosis. And in telophase is where they uncondense or decondense, and the chromosomes spread out into chromatin again, so they're the loose DNA fibers again. The nuclear envelope will form around each of the chromosomes, each of the clusters of the chromosomes. Okay, so it kind of looks like 2 cells already. That spindle breaks apart, starts to break down. The nucleolus will become visible. And we're getting ready for the cells to actually split and become 2 daughter cells. So we go into what is called cytokinesis. So how do they split apart after mitosis? Well, first of all, cytokinesis is the division of the cytoplasm, and is a little different in animal and plant cells. In animal cells, it forms what's called a cleavage furrow, and the cell membrane is drawn in until the cytoplasm is pinched into 2 parts. And each part will have its own organelles, obviously its own nucleus, and you'll end up with what are called 2 daughter cells. Now in plant cells, the cell membrane is not flexible enough to draw inward, so it can't make that little furrow. Because remember, it's got a cell wall, right? Well, what they have is it forms a cell plate in the middle there, and that cell plate is what will develop into the cell membrane. And then the cell wall will form in between the 2 new membranes. Now here's a little review of the stages of the cell cycle. So we had, the whole cell cycle, remember we had interphase and then we had M phase. So in the interphase we had G1, S, and then G2, and then we go into M phase, which was mitosis and cytokinesis.